Our next speaker is Dr. Eric Lai, who previously served as the Senior Vice President and Head of Genomic Medicine at Takeda Pharmaceuticals before his retirement in 2018. But the pandemic brought Dr. Lai out of retirement, and frankly, we're not letting him going back. But he is rocking it as the team scientific and clinical lead for the independent test assessment program. So over to him to let him tell you more about the expert HCV ITAP journey.
Thank you, Nate. First of all, I have to apologize that my computer keep telling me that my internet is unstable. I actually miss most of Dr. Collins' video because of that. I am going to turn off my video so that hopefully my audio will still hold. So I was supposed to do this presentation with Pam Miller, who is the ITAP program manager, but she has a last minute family conflict. So I'm going to do the presentation. Move. Next slide, please. Thank you. So Dr. Trombert review briefly the ITAP process. And in this slide, we are showing the ITAP process, how it is applied to the HCV program. Now, the critical component of the ITAP process are shown in this slide. The technology de-risking phase, which is shown at the top, by the Emory University and the Grady Hospital test validation team was really critical to the planning and success of the project. The validation team confirmed the LOD very quickly of the assay that we were going to use, and the Grady Hospital performed a clinical study in a very short time. In a month, they were able to enroll over 100 subjects so that we have very good confidence of the performance of the test and provided critical information on how to prepare for the clinical trial. In addition, the Emory testing lab also provided the fingerstick analytical data for the actual FDA submission. Now, that's a first for ITAP due to the complexity of the project and also the IRB limitation on some of the testing procedures that cannot be performed by CRO. Now, the urgency and the speed of the project were listed at the bottom of the slide. It took us 4 months with 15 sites to enroll 1,200 subjects. That is typically done similar to a phase 3 trial of a drug trial and usually takes about a year. That was a lot of coordination. And the submission of the de novo from April 15 and get market authorization in June 29 took 73 days. And it's really a process that usually takes easily 6 months. And I have to give a shout out to Wei Jiang, the regulatory manager at Cephia, who was masterful in organizing the submission. As a result of all of the work of everybody, we completed the project under budget and 1 day ahead of schedule. Next slide, please. So what were some of the key success factors of this project? We have outlined it in this slide, and we highlighted some of the important points up in the top, especially applied to the HCV project. First, it was very clearly right from the top, the visions of all of the leaders from the different agencies, all the way from Dr. Collins to Dr. Trombert and CDC and FDA. Everybody was right on board in the beginning, and everybody had the same vision. And then the project team was empowered to accomplish the goal set up by the leadership. At no time did the leadership involve themselves with the operational aspect of the project team. They basically set the vision and say, go ahead and do it. Now, that's great. We have a clear lines of decision making and authorization, and it will share the urgency around all the stakeholders that we get involved. To give you an example of the hands-off um, 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 operation from the leaders, I remember at the second monthly update when we reported that we had 5 positive subjects at month 2 of the clinical trial, and we need 120 for the whole trial. And Dr. Collins asked, how are we going to achieve the goal when we have almost 50% of the timeline of the clinical trial already gone by? Well, we did not let him see us sweat, but on the other hand, he also trusts us to carry out a plan that we assure him that we will be able to carry out. The important part is that we apply a proven process that we have carried out and performed with COVID, with the COVID flu multiplex, with monkeypox, and at that time we had over 20 EUA and projects authorized. The other thing that's important is that we have selected very carefully the sponsor for the project. And we have a very good comfort level that we will have a high probability of success. And we engage all of the teams that are highly specialized in what they do. And our collaboration with the FDA, as stated by Dr. Trumbert, was absolutely critical. And all of the government agencies help us when we need help. Next slide, please. Now, what were some of the key challenges with this project? The first one I mentioned is that we had to manage a large number of clinical sites over a 4-month period. Usually, this would have taken us easily 12 months or more. That was a major work. And the other challenge was that there was no 510K clear sample collection device for fingerstick. So we need to develop a lot of training material for that blood collection process in order to make it happen. We also have difficulties in procuring some of these materials because there was a lot of materials and a lot of them, it's all over, not just in the United States, but in a different part of the world. The timing and communication across many of the teams were also a challenge because in the US, we have people from the East Coast all the way to Hawaii. And then we have the Swedish group in Sofia and South Africa. So any meetings that we have, somebody is going to be up either at 3 o'clock in the morning or going to bed at 3 o'clock in the morning. But we did it. We made it happen. We also have very, very fast identification of the clinical site, which I will mention in the next slide. And we have been able to do all of this in a very short time. So fast that some of our key challenges was actually in documenting some of the data and documentation issue often show up on the clinical path. And this is something that we have learned for our next project. Next slide, please. Now, communication, as I said, is really critical. We decided to divide our communication team so that it's clearly technical leadership execution. And we assign tasks to specific individuals, not groups, and we have very efficient hands-off. So, for example, dealing with the FDA, we have a minimum of twice a week calls with them, and I will have emails with them almost every day. And then every Friday at 3:15 in the afternoon, I will have a discussion phone call with Ennis in the FDA, and we go through the whole week as far as what we have gone through, what we need to do next week, and so forth. The collaboration and the handoff with the FDA was absolutely critical with all aspect of the project and throughout the whole project. And at the end, we all say that it is the people that makes it work, not the technology. Next slide, please. Now, there are some things that we have done with the HCV project that we have never done before in a prior ITAP program. The first one is the groundwork for identifying all of the clinical sites. We did extensive surveys. We engage networks that have a lot of information on the HCV principal investigators. We went into literature. We talked to the CRO so that we very quickly identify a lot of the clinical sites that were suitable for the HCV projects. And luckily for us, that Dr. Jennifer Haven at Kentucky have done a study using the device that subsequently ended up to being the de novo. Very big study that we can learn a lot of the information from. Now, we also, based on my former pharma training, realized that because of the size and the speed of the trial, we really have to have a dedicated clinical trial oversight team and person within ITAP to facilitate all of that work in the clinical trial. And that was something that we've never done before. And the last part, as I said before, Emory actually contribute significantly to the analytical data for the submission. And that is also something that we've never done before. Next slide, please. Now, what are some of the lessons learned? The first one I have to say is that we try really, really hard to control the scope creep. Now, during the whole project, there were a lot of wouldn't be nice to have. The problem is that every nice to have would translate into 1 to 2 months of additional work. Now, that would be fine for a project that lasts 18 to 24 months because you can try to catch up, but not for this project. So we tried really, really hard to control that. The other thing is that while we thought that a CE mark product would be easier to adapt to the US, there was still significant adaptation and de-risking needed. And the third point was actually a really surprise for us. We thought that the blood collection would be challenging for a lot of people that inject drugs because they might have callous fingers for finger sticks. It turns out that the venous blood drawn in these patients, in these subjects, were actually more difficult than having finger sticks. We have more people that we end up not be able to get a venous blood drawn than the finger stick. So this showed the importance of the point of care finger stick assays that we have developed. Finally, a lot of the long lead time for acquiring some of these items from overseas require constant pushing from multiple agencies. And that is something that we have learned, and we are going to apply our learning to the HBV projects. Next slide, please. Now, with such a fun project in such a short time, we work very well, but then also there are times that we have interesting directive from the different people that runs the project. I remember that when we first started doing the project, we tried really hard to have it as quickly as possible. And at the first meeting with Dr. Collins, we told him that we can do it in 12 months. And the first thing that came back to us was, can you do it 3 months faster? And that's why we end up at the end of June. And when we talked to the FDA, Uwe was very, very sure. And he said, when in doubt, escalate it to us. And when we were doing our final submission, and we were working really hard on getting the de novo through by the end of June, there were discussion about, well, can we push it into July? And Innes from the FDA was very clear. There's no July. We have to get this done in June. And finally, because of all of the different parties involved and all the different reagents that we have to make sure that we ship to different parts and so forth, we actually have some of the reagents shipped to people's home over the weekend in order not to lose the weekend. And they were always saying that, can we please confirm that we are shipping this correctly? So those are some of the fun things that we learn in the project. Next slide, please. Now, at the end, it really takes a village to get this done. And a lot of people support this project. Just like a film credit, everyone listed here play a major role in the success of this project. And yes, there were more FDA folks on the list than CDC, NIH, and ITAP. And that is correct. FDA really put in a tremendous effort in this project. And I can tell you, every single one of them contribute significantly to the success of this project. And with that, I close and give it back to you, Nate.